Our next speaker is Laura Rozek. Laura and Dana were recruited as a pair of epigenetics superstars, but we didn't have to go far for Laura. We crossed the street, nabbed her from the otolaryngology department. Please welcome Laura Rozek.
Can you believe I have to follow the polar bear pictures? Thanks, Andrew. Real nice. But I think, oops, I'm going to go backwards. I don't want to give it away. I think that I really am a scientist in the best time to be a scientist. The Human Genome Project started the year I graduated from high school, so you can figure out my age. By the time I made my way to graduate school in the late 90s, we were already well underway in unraveling the human genome. By 2003, we had the complete sequence. I mean, how amazing and incredible is that? Not only was it this time where we thought that we were going to be able to unravel diseases and understand risk for disease and prevent them, but it was also this amazing time of collaboration. Every one of my mentors throughout my graduate school time told me that it was an unprecedented time of groups coming together to try to solve a problem that they thought would better humanity. So again, I think it was just the best time for me to grow up as a scientist. And this, from data that came out in 2009, right when I was starting my position here in environmental health sciences, the Center for Health Statistics released these data of about 60 years of mortality rates in the United States. On the y-axis, you could see the number of deaths per 100,000, and the x-axis, the decades. And, you know, a lot of us are scientists here. Don't need to be a scientist to see some of the really striking things coming from these data. So nice job, people who study heart disease. I mean, the mortality rates from heart disease have just plummeted. Prevention measures, including pharmaceutical and statin and all sorts of interventions have really stopped this. So, you know, good job. But wow, what's up with cancer? How many people here are surprised by this? The mortality rates from cancer were basically the same throughout this entire period. I mean, we're talking smoking rates had gone down. I mean, evidently, you know, all these things we've been told to do, you know, what's causing cancer hasn't helped. Mortality rates are about the same. And if as public health professionals we judge our ability to prevent disease and mortality rates, frankly, we're losing the war on cancer right now. People are still dying from cancer at the same rates, and that's just simply unacceptable here in the United States and worldwide. Most cancer researchers kind of took this as a cause to go out and race for the cure, find the cure. You know, let's go out and come up with better therapeutics for people who get cancer. Undoubtedly, very, very important. I work with surgeons over in otolaryngology and radiation oncologists who are doing an incredible job coming up with new therapies that are less toxic to the body, where people can go on and live with cancer. And then some cancer researchers say cancer is now a chronic disease, that we can treat this as a chronic disease through the life. I think, again, as public health professionals, when we look at this, we have to think to ourselves, wow, you can't die from cancer if you don't get cancer. I mean, it's a simple fact. One of the things that we can do as public health professionals is to try to better understand the causes of cancer. Because I don't think we really know what causes cancer all that much. I don't think we know the risks in populations, and I think we could do a better job of designing studies to better understand it. So throughout time, you know, man has always wondered, what causes disease? And if you talk to any physicians, any of the cancer doctors I work with, one of the first questions the patients say is, why did I get cancer? Hippocrates tried to describe this through describing humors. 4 body fluids, black bile, yellow bile, blood, and phlegm, and the balance of these humors determine whether you were healthy or unhealthy. Galen, the Greek physician, looked at these different imbalances of humors and tried to assign different diseases based on which humor was out of balance. So inflammation was simply when your blood was out of balance. But the one thing, the really nasty black humor, the black bile, was reserved for only 2 diseases. It was reserved for depression, and it was reserved for cancer. What causes cancer? Black bile without boiling causes cancer. Yeah, it is kind of funny. What's even more funny is to come up to 2000. So 2,000 years later, we put men on the moon. We've developed Twinkies. I love Twinkies. I know they're going out of style. But 2,000 years later, we have so much more information. So let's think about it. Let's ask We asked Mel Graves, who is one of our landmark British physicians who studied leukemia, so what's causing cancer? It's the year 2000. It's bad bile, it's bad habits, bad bosses, and it's bad genes. We sure haven't come very far, have we? I'm pretty sure they had bad bosses back in Greece, right? I mean, lots of people died, things like that. So again, just saying, I don't think we've really come all that far. And I think we have a pretty big charge, again, as public health professionals to try to understand this. So what do we know about cancer? The Human Genome Project, again, how many times have we talked about it today? We could talk about it until the end of time. The contributions have been numerous. One of the big contributions is that we have a stronger understanding of the cancer genome. So we now know a lot of the somatic mutation changes in different cancers and how they relate to different cancers, either between cancer sites in the body or between individuals. This has been very useful for treatment because we know different somatic mutations in different genes, determine whether or not a tumor is more or less susceptible to chemotherapy. We also know a lot of these changes are associated with different environmental and other exposures. So we know that. We also know that cancer takes decades to develop. You're not going to start smoking at 50 probably and develop your cancer. But all you people in your 20s out there, all my students, I wouldn't start smoking now because that's what's going to cause cancer later in life. So we know an awful lot about the cancer genome. But what do we need to know? We need to know how toxins, how the diet, how things in the environment all aggregate together to cause cancer. So I think Dr. Meeker did a really nice job talking about mixtures. Most of our models of determining whether or not a chemical or any kind of pollutant is toxic is by looking at the single pollutant in an in vitro or animal model to determine whether or not it's mutagenic. This is simply not adequate because we know that every day we're exposed to an aggregate of chemicals, and this is unavoidable. There is no way, as John said, we're going to get up in the morning and say, I'm not going to have any endocrine disruptors today. And I can tell you, I know we're all exposed to bisphenol A, and that's going to be something that's going to happen for a long time. So one of the things we need is to be able to understand these chemicals, understand their carcinogenic potential in humans. Now, I'm a molecular epidemiologist, so my bias is to use population studies. Population studies, I think, are a very good way to look at people in a natural environment, in their own natural experiment, to see what causes cancer. So I study people. Essentially, I study people, identify those who have cancer, and compare people who have cancer and who don't have cancer. But a good question here is, how can we infer causality? By just looking at people who have cancer, who don't have cancer, asking them questions, maybe measuring a few things. In fact, we can do that. One of the seminal studies, the Dahl and Hill study that determined that smoking caused cancer, didn't even set out to think that smoking caused cancer. Richard Dahl went out every afternoon, very famously, to have a cigarette sitting outside of his lab. But they looked at cases and controls of lung cancer in London and asked them a whole lot of questions. Varied in the questions about how much fried fish they ate every day or how close they were to a coal smelter was one simple question that just asked them, do you smoke? When they sat down and they analyzed the data, unequivocally, smoking caused cancer. Richard Dahl immediately quit smoking. That's not even a joke, that's a true story. But we're limited as we start to think about these kinds of studies in the world we live in now, because that was a really obvious exposure. That was an exposure that had a large attributable risk. So how do we study exposures that may have a smaller risk for cancer? And how are we able to infer causality from this? Most people would say you can't. But I'm going to put up another quote from Richard Dahl, which I'm assuming he made after he stopped smoking, that whether epidemiology alone by itself in strict logic can never prove causality may be questioned. But the same must be said of laboratory experiments on animals. And I say this because I think we need to integrate. So, as Howard mentioned, I share a lab with Dana, Illinois. We think a lot about the ways we can integrate different types of information from animal studies and from human studies, and from the human genome and information we've gained from that to infer causality in population studies of carcinogens. Very simply, what we can do is when we look at cases and controls, or people who have disease and don't have disease, is that we can look at their DNA. We can look at changes in their DNA that we think are associated with disease. And by that, we can say that this causes the disease, and think of it as a carcinogen. Most models we think of whether or not a toxin causes cancer, or whether or not they're mutagenic, whether or not they cause a change in DNA that truncates the gene, so it's not expressed in some kind of, very important cell cycle gene. But any of you who heard Dana's talk and aren't excited about epigenetics must have slept through it, because one of the things that we know is that these toxins may cause epigenetic changes and then influence the risk of cancer. Why this is so exciting is because we know that these changes are reversible. We know that they can be changed through diet and through lifestyle changes over the lifespan. As I said, cancer takes decades to develop. If in some way we could determine that a toxin is associated with epigenetic changes that may lead to disease, not only can we determine that that toxin is carcinogenic, but we also may be able to reverse it. I'll give you an example of a study that we're starting on very early right now in my laboratory. There's high levels of cadmium in Thailand. And people who live in areas where there are high level of cadmium in Thailand have higher rates of cancer. Really, really, really interesting correlation. But how do you prove causation? Cadmium in and of itself is not mutagenic. It's really not all that bad for you. But one thing that we can do is we can look at people in this region who have cancer, look at their levels of cadmium, and look for epigenetic changes in their tumor or in their circulating DNA. Then we can also look at the controls and try to correlate cadmium as a cause for cancer. Not only is this important to show that cadmium may be carcinogenic, but it's important to know the level. I mean, there may be levels of cadmium that are carcinogenic, but then at lower levels, you're fine. You can go through life, live in Thailand, which is pretty nice, by the way, and then you can go along your merry way. And I think that's really an important thing, again, that if we haven't emphasized it, that we can. Part of environmental health is knowing the thresholds and what we can live in, because a lot of these exposures are ubiquitous, and a lot of these exposures are unavoidable. I'll close by talking about the global burden of cancer and the global burden of cancer mortality. Well, I emphasized the United States mortality rates from cancer early on. When we look worldwide at cancer mortality rates, the vast majority of the burden are on low and middle income countries. This makes sense. Anybody out there would say, well, that's where most of our world sadly lives. But this difference is going to be exacerbated by the year 2030, where over 80% of the cancer deaths are going to occur in low and middle income countries. This is particularly frightening when we think of how much cancer care costs. Again, I was in Thailand last week, and I was lucky enough to give a talk at their National Cancer Institute meeting. And I complimented their organizers on some of their presentations. I said, wow, half of your talks are about cancer prevention. This is amazing. You certainly don't see that in America. And he looked at me and he said, we can't afford to treat cancer here. So in America, where cancer is just, maybe something we can't afford to treat cancers, we can't afford the chemotherapies, do we want to spend our money on it? They don't have the money in these countries to treat cancer. Prevention is their only option. Now again, to be anecdotal, I was at a study section this week where somebody actually said, it's impossible to study the environment in cancer. We simply don't have the money to look at toxins and different types of cancer. It's impossible. And I'd say that every great success story started with somebody saying it was impossible. And if we took the resources that we put into something like the Human Genome Project or the Epigenome Project, I think we could understand how complex mixtures of chemicals are associated with cancer and come up with preventative measures. And with that, thank you.